Hello students and welcome to my review video on DNA. In this unit we've covered DNA, the experiments that led to the discovery of DNA as the genetic material. We then covered DNA structure and how it leads to its function. We also covered DNA replication or how it's copied. And then finally the central dogma or going from DNA to protein, DNA to RNA to proteins. Just like all my videos, this is meant to help students. It is not for profit. Nobody can derive money from this. It's just meant to help. Many of the pictures come from Khan Academy, and that is available for free at khanacademy.org. Like all my videos, it's best to pause them, to ask yourself questions, and then come in, TASM students, and ask me questions and work together in study groups as you work to master these learning targets. The learning targets come from these standards, and here they are. You can pause the video and read them and figure out, have you been meeting these? Are you able to ask questions about DNA and chromosomes and coding? I've had some really great conversations with some of our students about what's going on. Why are we, a mixture of our parents and our grandparents, etc. We're going to get more into genetics after CISA and after this unit. Can you construct an explanation for how DNA determines the structure of proteins? And so this we've really worked hard on in class, and I'm very proud of you for learning so quickly how to use, how to turn DNA into RNA, and then how to use the codon wheel to turn RNA into a protein. All right. We, like all of our units, have an underlying phenomena. And so in this one, we watched the video about David Vetter, the boy in the bubble. He unfortunately passed away before he got to be a teenager. They've named an elementary school after him in Houston, Texas. He was born with a compromised immune system due to SCID, or severe combined immunodeficiency syndrome, I believe. And it was due to an error in the protein synthesis, right? And so it's a real life example about how a mutation in DNA may lead to something harmful. Mutations in DNA can also lead to something beneficial. It can be selected upon by natural selection. We'll get more into the inheritance pattern of SCID here. I told you in class that there's an X-linked SCID variant, and that's the one that David got here. X-linked diseases tend to affect men more often, such as color blindness or baldness or hemophilia. There's also an autosomal recessive version of SCID that you got to work on with the gizmo. And unfortunately, it was pretty disappointing that the gizmo didn't work very well, especially for B block. If you have time, I encourage you to finish that gizmo. And I think that would be an excellent way to study. And it's really illuminating, right, to see the real world problems that might happen with a mutation. All right. I gave you a story about the discovery and structure and function of DNA. We'll go over that in just a minute. You do not need to memorize any of that, but it was an ongoing process. And it was finally cracked and published in Nature in 1953 by Watson and Crick. And then I told you all about Rosalind Franklin and some of the injustices there. Remember that in the beginning with these experiments that I'm about to go over, these 3 experiments, the big question, students, was, is it protein or DNA on the chromosomes that are genes? And DNA has 4 bases, right? You remember A's, T's, G's, and C's. And there are 20 different amino acids that make up proteins. So kind of like all the letters in this sentence, there's 26 letters in the English alphabet. They can make up lots and lots of words in different combinations to make big books. So a lot of people thought it was proteins that held the genetic code. DNA just has 4 letters. So how could that be complex enough to code for everything? Let's dive in. Here is the first experiment. What I would like for you to do is to pause the video, and can you come up with, what did a scientist figure out? This is Frederick Griffith. And then what's the reasoning going on here? You could even use a claim evidence reasoning framework to help you. So go ahead and pause that video. I'll put this back up. Okay, hopefully you got something along the lines of, the claim is that bacteria can be transformed or changed. The evidence was you put that heat-killed S strain with the non-harmful R strain, and it changed it. It gave it its properties, right? And so in class, I talked about maybe you're out on Discover Oman, and if you got stranded, you might need some water, and hopefully you would heat up that water to boil it and kill any microbes in it. Well, that's what he did here. He heated up the S strain and he killed it, but it still turned the R strain into a killer one. What we know now is that R strain was able to pick up the DNA from the S strain. Their reasoning, they didn't know why, but we call that transformation of bacteria. And just a little shout out here, now that we're in the middle of Discover TASM, is that you get to do bacterial transformation if you take AP biology with me. So here's the graphic, and I think it's easier to see and to interpret with the graphic. Remember, I will give you the graphic on the test. Here, you put that rough strain into the mouse. Mouse is fine. The immune system's able to fight it off. Put the smooth strain into the mouse, and the mouse dies. The reason being is this smooth strain has a coat on it that helps the smooth strain evade the immune system. You take that same smooth strain, just like I talked about being on DO, heat it up, and it does not kill the mouse. But if you take the heated up strain and then put it with an R strain, it transforms the R strain. It makes that rough strain deadly, essentially turning it into a new smooth strain. All right. So from the Griffith experiment, Avery, McCarty, and McLeod came along and said, let's try and figure out, is it DNA or protein that's causing the transformation of the cells? And this is such a beautiful experiment, the way they set it up. If you can understand it, then you really got a good grasp on experimental design. They took the heat-killed S cells. Let's go back. Remember this one, these heat-killed cells. If you put it into the mouse lives. And what they did was they isolated different factors. So they took out all fats. They took out all sugars. That's what lipids are. Lipids are fats. And what's left are proteins, RNA, and DNA. And then one at a time, they removed either the proteins, the RNA, and the DNA. So here they removed the protein, all right? And so I might ask you on your assessment, if they remove the protein, will the R cells become deadly? What's going to happen to them? The answer is yes. We now know that DNA is a transforming factor. So if you take away proteins, DNA is still there. DNA and RNA are still present in this culture, and it's able to transform the cells. In this test tube, we're going to take away RNA. We're going to use an enzyme to chop it up. When we take away the RNA, the R cells also still become deadly, right? And so the DNA is still present, and it was able to transform the R cells. Here, this is the beauty of it. When you take away the DNA, the R cells are not transformed. So the absence of DNA causes these ones to not be transformed. And from that, you can assume or claim that DNA is a transforming factor. Maybe there's something else perturbing the experiment, but it's a good setup, and it gave us a good background for the next experiment that's going to go on with Hershey and Chase. So this is a tough one, and it's a tough one every year for students. So what I encourage you to do is to pause the video and to try and go through claim evidence reasoning with this graphic, all right? And then you can check yourself with my next slide. Okay. Here are my answers for claim evidence reasoning. You can test yourself and see if you got it, right? And so when the DNA was disabled, the R strain was not transformed. All right, let's go on. The Hershey and Chase experiment's coming up next. This is another beautiful one. It's often called the blender experiment. And they dealt with viruses, and they knew that sulfur is an element that is found in proteins but not DNA. And they knew that phosphorus is an element that's found in DNA but not found in proteins. And so from this, they were able to do a really cool experiment. So here we go. These viruses, they're called bacteriophages because they infect bacteria, were grown in either radioactive sulfur, as shown here in pink, or in radioactive phosphorus, as shown here in blue. The ones that were grown in the radioactive sulfur, remember that sulfur is found only in proteins, or not only in proteins, it's found in proteins and not found in DNA. So these ones have pink proteins. This one has radioactive phosphorus, which is found in DNA in the sugar phosphate backbone, but not found in proteins. So these have blue DNA. You can think of it that way. They let the bacteriophages infect the cells, and they reasoned that These viruses are going to infect the cells with their genetic material to make more copies of themselves with the DNA. They then spun the system around really fast. And what that does is it puts all the cells at the bottom of the tube. It's like if you trolled around a bucket of water really fast, all the water's at the bottom of the bucket, it stays in there. And so then they looked at the bottom of this tube to see what was in the pellet. And they found that the radioactive phosphorus was in the pellet, which means that that's what was injected into the cells. The radioactive sulfur was found in the water above. And so that means that the viral parts that were still around are up here in the water, that it was not infecting the cell. The cell is heavier, it's bigger than these bacteriophages. So that's why it's at the bottom. A pretty cool and beautiful experiment. Unfortunately, only Hershey really got recognized for it. Chase did not. Another injustice. All right, you can pause the video and do claim evidence reasoning for Hershey Chase. Here are my answers. Then you can check yourself with. Okay, we did a discussion about the structure and function and the search for it and who had evidence, etc. I don't expect you to memorize anything from it, but I tried to show you that not everything's in your textbook, right? And that Rosalind Franklin had a picture stolen from her, and more than just this picture, also some of her progress reports and writings were stolen from her and shown to James Watson and by virtue of Francis Crick. And then I also showed you that the only one that's still alive is Watson. He's still alive today, and that he's racist. He has poor views, things that are terrible, right? And there's he's a genius. He really did a lot of work with DNA, but he's lost all of his titles, and nobody really wants to associate with him anymore because of his racism, and he also makes sexist remarks. And I would encourage you to pause and read this quote by Adam Rutherford, a editor in Nature, and just great scientists are not always great people, and I want you to be a great person first and foremost. And so that's really important to me, that you think about embracing diversity, and that diversity makes our school and makes our world a much stronger place, right? It is better to hear opinions that differ from yours, to hang out with people that look different from you and that may have different beliefs, and to learn from them. All right, let's bring it back here. So at this time, we now have DNA as the genetic material, right? And so we see it all the time. It's used in ads everywhere. Progress is in its DNA. We got the anti-aging DNA perfume at duty free. So how would you describe DNA to a 4-year-old, to a 15-year-old, to me, your biology teacher, now that you've had a couple of lessons on it? Can you pause the video? All right, I'm curious to hear what you have to say, and I'd love to hear it in class. So in biochemistry, we often say the structure of a molecule leads to its function. So the structure of the tennis racket enables it to be a good way to play tennis, right? Way to go slice of tennis. The structure of these goalie gloves leads to them being sticky and helping to catch the ball. Structure of the track cleats leads to them helping you run fast. Structure of a phone enables it to do all the things you love with your iPhone. The structure of the basketball hoop enables it to be a basketball goal. So the structure of a molecule leads to its function. Let's start with that second part. What is the function of DNA? And so go ahead and pause the video and discuss that with your parents or with your siblings or with your friends. Hopefully you got something along the lines of serving as the genetic material in the cell, carrying heredity through generations. It helps to code for proteins. And that you might remember that the inherent mutability of DNA provides the raw material for evolution, right? That as DNA gets mutated or changed, sometimes that can be bad, sometimes that can be good, and that can be acted upon by natural selection. So let's see how the structure of DNA leads to those functions. Remember, genetic material, proteins, evolution. Well, it stands for deoxyribonucleic acid, just so you know the name of it. It's found in the nucleus of the cell, and it makes up the classic chromosome structure here that looks like an X. It's really DNA kind of wrapped around some proteins. Here's just another slide showing it. So it's inside the nucleus of the cell, and you probably learned about the cell in middle school, and we'll go over cellular biology more in about March this year. So it's the blueprint of life. The sequences of these bases is what leads to us. And so we watched a quick TED clip with Professor Ricardo about where he had David Vetter sequenced. He had a human sequence, and he brought out all these books, right? And he lined them up that showed this is what his DNA would be if you typed it out. And it was like 10-point font, and it really stirred up some talks in class. And so if this were, the DNA of me, John Gibney, and the DNA of my wife and kids, or my friends at home, right? The differences between us is probably just like a few pages, just about this amount. All of this is the same through humans and is conserved. And that's because we all have the same genetic makeup for metabolizing sugars, for making insulin, for making human growth hormone, for other purposes, right? Our phenotypic differences that you might see, like eye color, hair color, or skin color, is just that small amount. So the sequence of these bases is what leads to our physical traits, right? And they're read and they're decoded by different enzymes. So let's see a little bit more of it. A gene is a segment of DNA, and it's going to code for a protein. Proteins determine how we look and our insides and everything. All right, so maybe this one would determine purple hair and this one determines yellow hair, just as an example. We're going to read DNA in sets of 3 letters, which you're very comfortable with now. All right, let's get a little bit more into it with describing how the structure of DNA leads to its function. Can you identify in here where there are strong bonds and where there are weak bonds and why? And so hopefully you get something along the lines of there are strong bonds in the backbone of DNA. Here it is. You can see that the backbone is the same on each side, but this side runs in one direction and this side runs in the other direction. But in essence, it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. What can differ is the order of the bases. These bases are held to, well, remember that these are strong bonds. These are covalent bonds over here between the phosphate and the sugar. They're very strong. They are sharing electrons. Here are, in the middle, are hydrogen bonds, and these are weaker between the bases. So that's interesting. Why would we have a weaker bond in our DNA? Shouldn't DNA be strong to hold everything we need? And by virtue of having weaker bonds here, it allows us to open up DNA to make copies of it and also to transcribe genes, translate them into proteins. So for instance, we could open this up right here, access a gene, and make something like an adrenaline protein or an insulin protein as well. A's are going to always bond with T, and G's are always going to bond with C's. And I told you one way you can remember that is you should put ashtrays in the garbage can. And so you should never smoke. It's just the arrangement of where hydrogens and then the different oxygen or nitrogen is on the other side that allows them to arrange this way. The A's and G's have 2 rings, and the T's and C's have 1. And these are called purines. These are called pyrimidines. Let's not give you too much information, but basically a 2-ring nitrogenous base has to pair with a 1-ring one, and that will help you have the helix structure. If you have 2 and 2, it wouldn't work out. It wouldn't be able to have that double helix structure. The double helix structure is important because enzymes can attach to the major groove here and open it up and transcribe genes. All right, I'm going to get my computer battery. Okay, I'm back with the charger. Not going to lose you. Let's move on. So you can repeat that. You can go through the structure leading to the function to code for our genes. So that it's embedded here, and these order of these bases is going to make you, you, and make you special. The strong backbone helps to keep it together inside the nucleus, and then the weaker hydrogen bonds allow it to be open and transcribed and translated. All right. A nucleotide is one phosphate, sugar, and base. That's important to know. That would be like a Lego. And then that one Lego, you put them together like the students did with the physics projects in the Commons, and you make a whole structure. So this would be a Lego, 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 Lego. We call those monomers. So it's a building block to make the polymer of DNA. DNA has a sugar that is C5H10O4. That's not important for you to know, but it's slightly different than the sugar that's in RNA. All right, let's move on. We've already been over these hydrogen bonds here. One thing that's interesting is you'll notice there's only 2 bonds between A's and T's, and there's 3 between C's and G's. So A's and T's are actually a little weaker than C's and G's. So many genes will start with A's and T's, so that way it's easier for the enzyme to open up the DNA and start transcribing and transcribing it then. And it's held together by those really strong covalent bonds. So here's just an example. This is called dehydration synthesis, when you take away a water molecule and then bond there. And so it's going to be bonded on that oxygen. It's very strong. You're not going to break it. Okay, the strands run in opposite directions. In class, I talked about salt and caboose highway, right, running in different directions, and that helps to lead to the double helix structure. When you take AP biology with me, like, you'll learn that we can only add to the 3' end. So it's going to lead to some interesting ways that DNA has to be copied, but that's a little bit beyond this course. And so let me stop doing that. Let me just stick to what you need to know. Okay, can you make the other side of this DNA molecule? Hopefully you can. You got something like this. In DNA, we have A's and T's, G's and C's, and A's always pair with T's, and G's always pair with C's. Likewise, T's with A's and C's with G's. Erwin Shargoff right here figured out that the amount of A's and T's go together in G's and C's, and so you can see some interesting differences between humans and other organisms. Can you try this problem, right? If the percentage of A is 30, what's the percentage of G? All right, if you do it, you know that if A is 30, that means T is also 30, so that's going to be 60. And it has to add up to 100, so 100 minus 60 is 40. Divide them between the two, that leaves 20 for C and 20 for G. All right. We've done a bit. We've done the experiments, and we've done structure and function of DNA. So what do we have left? We got left replication, transcription, translation, and mutation. So if you need a break, now's a good time. If you're ready, let's move on to DNA replication. So we have to be able to make copies of DNA, right? Every time we make a new cell, it needs to have a whole copy of the DNA, so that way it can properly code for proteins and do the work it needs to do. So right now in my body, I'm making new cells. Most cells divide on average once a day, except for some brain cells and other stuff. And so cells in my intestine, I'm replacing them right now as I make this video. So how do I do it? Replication means to make a copy. And so I'm going to ask you, why is it semi-conservative? What does that mean? Why don't you pause the video and see if you can draw it out on a piece of paper? You're going to be constructing your own model here, right? And you should use some DNA letters to help prove it. All right, let's do it real quick here. This is the one I used in class. So I got up on the board and we drew it out. We added letters. And then when we separated it with an enzyme called helicase, and then we had another enzyme, DNA polymerase, that added the new nucleotides, we saw that by virtue of the base pairing rules, because A's go with T's and G's go with C's, these two strands will be identical as long as you had one old template strand for the creation of the new strand. Let's see it here with some real letters. And so here is the DNA strand. We're going to split it up. So you see you got C, T, A, A, G, C, C, T, G, A. That was from that top strand. Down here, we'll take the G, A, T, T, C, G, G, A, C, T. And due to the base pairing rules, because G's always go with C's and T's always go with A's, it should be identical when the new strand is added to it. And that new strand is called the daughter strand. There can be errors. DNA polymerase works very fast. It works like a jet engine at about that kind of speed. So if there are errors, they're hopefully caught by proofreading enzymes. They can be chopped out and fixed. They can be fixed right there at the site of synthesis. But occasionally errors will get through. And if those errors happen in a region called a proto-oncogene, that could turn it into an oncogene, which you might have heard of the word oncology, and that could be cancerous. It'd be like if a gas pedal gets stuck down, it makes a cell divide too fast, right? There are large sections of DNA that don't code for proteins. I told you in class we used to call them junk DNA, but we now know they're really important. So the term junk DNA is silly. And if a mutation happens in there, it might not affect gene, and it might not affect a cell to become cancerous. So also, cells check themselves all the time. They have genes that say, hey, like there's a famous one called P53 that checks the cell, make sure it's not cancerous. When it is, it might say, we need to get rid of this one. All right. I got a little long-winded there. Sorry. Can you remember from class helicase and DNA polymerase? These are enzymes. They end in ASC. That's just a trick to remember that denotes that they're enzymes. Enzymes are proteins. So these are coded for by segments of DNA. DNA helicase is an enzyme that's going to unzip or break the DNA molecule so that way it can be copied, right? The area of which it is breaking it is called the replication fork. And in DNA polymerase is going to be this enzyme that's going to add new nucleotides. All right, so here's a real nice picture of it. And so it's going to add new nucleotides. All right, so here's the steps. You could pause the video and read it. I'm not going to read it to you about what's going on here. In eukaryotic cells, that'd be plant and animal cells, this is happening at hundreds of places on a DNA molecule, so that way we can get it all done. The real DNA, when it's being copied, kind of looks like a bowl of spaghetti. Right? It only comes together during that classic chromosome structure when a cell is dividing and when it needs to split its DNA equally. Here would be an example of eukaryotic DNA. So this would be for humans, right? Lots of spots where that DNA synthesis is occurring, a DNA replication, excuse me. And then here in bacteria, they just have one long strand of DNA that's circular. And so it'll go from one end to the other end and make a copy. Bacteria can also have little extra bits of DNA that exist in it called plasmids that are little circles that can also code for proteins. We did not get to this year, which was a bummer. It's a super cool experiment. And so I think I might do it during CISO while some students are gone. That way they don't miss anything that'll mess them up for genetics. So we'll come back to Messelson and Stahl, and we'll do claim evidence reasoning with it, and you'll get to act it out. I think that'll be pretty cool. Okay, that was DNA replication. So we've done structure, function, replication. So now we're on to transcription and translation and mutations. So let's dive in. So what's the big picture of protein synthesis? Hopefully you got something along the lines of DNA, RNA, protein. And I told you the story that as I got interviewed here at TASM, that was one of the questions that they gave me. They're like, describe protein synthesis to somebody that is not a science person. And so I talked about how, well, DNA would be like the cookbook, grandma's cookbook. We're going to take the recipe, which is going to be the RNA, to the store. And at the store, we're going to get all those amino acids, the ingredients of the meal. And then at the ribosome, we'll make the protein, and that protein would be the final meal. All right, can you compare and contrast transcription and translation? Transcription is going to be the coding of DNA into RNA. And then translation is going from RNA to protein. Transcription takes place inside the nucleus, and it involves an enzyme called RNA polymerase. This is easy to remember because it's making RNA. Translation happens outside of the nucleus in the cytoplasm or attached to what's called the endoplasmic reticulum at the site called a ribosome. And everything that's alive has ribosomes to make proteins. Here's an example of RNA polymerase at work on a gene. So it's reading it and it's making the mRNA transcript. You'll notice a couple of things here. It's single-stranded, whereas DNA is double-stranded. And it has a U instead of the letter T. it uses uracil instead of thymine. This helps to differentiate RNA from DNA. One way you can remember this, I believe, oh, I'm sorry, I thought it was up next. But you can remember it's almost Valentine's Day, 11 days away from the recording of this video, and RNA loves you. All right. So genes are DNA instructions. They control the production of proteins. The DNA remains in a nucleus, right? It'd be like me. I'm not supposed to leave the classroom. I might send you a messenger to Mrs. Rita to help out to do something. Okay. So some properties of RNA that we just went over in the following slide. It's single-stranded. It contains a different sugar. It's called ribose. It has one more oxygen than DNA. It contains uracil, not thymine. One interesting bit, because it's single-stranded, it can kind of fold back on itself, and that's going to make a tRNA molecule later on. So can you transcribe the DNA here, ATA, GCG, into RNA? Always remember, RNA loves you. And the answer here was UAUCGC. So there's my RNA loves you. C to G, A to U. It's the messenger carrying the transcript to the ribosomes. All right, can you pause it and think about the three types of RNA real quick? Okay, here we go. We got messenger RNA. That was the one that we made. It was the transcript going from the DNA to the ribosomes. It looks like this. We have rRNA, or ribosomal RNA, that's going to help make up ribosomes. So that's going to be made, it's going to be part of the ribosome along with a little bit of protein. And in tRNA is going to carry the amino acids, and it's called transfer RNA. So you can think about it transferring the amino acids. Each tRNA is going to have a specific anticodon at its bottom, and it will carry a specific amino acid, and there's 20 of them. All right, so let's go through it again about how it makes a protein. It's going to transcribe, RNA polymerase is going to transcribe a gene that's going to make messenger RNA. Messenger RNA leaves the cell nucleus through a nuclear pore, which is a hole in the nuclear membrane. The messenger RNA then goes into the cytoplasm, which is the jelly-like substance inside of a cell, and it goes to a ribosome and starts. It is read, and a protein is made. A protein is many amino acids put together. Let's go through translation. And so here's a big picture view of it. The tRNA is going to come into the ribosome. It's going to match up with the mRNA, the codon, a set of 3 nucleotides, and it's going to bring its amino acid. And each tRNA has a specific amino acid. Here is a good picture of it. Here we would have AGU. We know that's RNA. It's got U in it. We can see that this is single-stranded, and this is going to be messenger RNA. AGU, this codon, is going to pair up with an anticodon, UCA. U is the opposite of A, C is the pair with G, and A is the pair with U. Notice how in tRNA we still use U's. There's no T's in tRNA, except for in the name right here. All right, this is going to bring a specific amino acid. Here's CGU. The codon pairs with GCA, the anticodon, that's also going to bring a specific amino acid. They form a covalent bond, very strong. They're going to share electrons. Now, these amino acids are bonded together, and they cannot break this. But just because they're bonded together, some of them might like other ones more than others. For example, this orange one might want to come hang out with this orange one. They have differing characteristics in what's called an R group. And so we acted it out in the hallway with kind of a role play. We'll get to that in a minute with mutations. Here's another example of it. So all proteins start with AUG. And so that's going to be the start codon, methionine. Sometimes if the protein doesn't need methionine, it might excise this out or cut it out later on. But anyway, when we're reading mRNA, we look for AUG to be the start of the protein. All right. Up next was GGA, and that is going to pair up with CCU, the anticodon, and it's going to bring glycine. So this protein is methionine, glycine, and then valine. Let's look at the chart. Here, let's look at AUG. Remember, we're going to go first letter, second letter, third letter. That's going to code for methionine, right? And so we had that here. Let's look at GGA. GGA codes for glycine. Here, this is interesting. So does GGC. That also codes for glycine. GGU also codes for glycine. So in this instance, this is a good example of the genetic code being redundant, right? These third letters right here can be can be U, C, A, or G, and they still code for glycine. So there's only 20 amino acids, but there's 64 combinations of 3 letters. So some of them have to be used over again. We call that the wobble effect, and we think that helps to mitigate or lessen the impact of certain mutations. All right. The way you read the chart is the same as you go first letters over here. And so if it's AUG, I'm going to look in this row. And second letters up top, so that's the column. So it goes row, column. So AUG, so I know it's here, and then I can just find the third letter, and it's going to code for MET. Remember that you can always write the 3-letter code on the test, and I'm going to give you the chart. You should never memorize this chart. There's no point to that. These are called mRNA codon tables. They are giving you the amino acid, and they're not dealing with anticodons. So you may wonder, why did I teach you anticodons? The point of the anticodon is that way the correct amino acid pairs with the correct codon, right? Otherwise, it'd just be all willy-nilly. But here, because it's CGU, it's going to only match with GCA. So you go to the right locker because you know your locker, right? And so here, same thing. Let's look up GUU on the table here so we can practice a little bit. G is in this row, U is in this column. GUU codes for valine. All right, that was a bit whirlwind, and I apologize. I didn't have as many questions in that part of the video. So pause the video. You have a lot of practice sheets on this. See if you can go through and work on protein synthesis, right? And so if you get to do the gizmo, you're going to get to practice this, and you'll get to see the protein being made. So what I did in class is I had you be a certain amino acid you were coded for, and then we went out in the hallway and we practiced folding, right? And so some amino acids like others. So can you describe how a mutation or a change in DNA can affect the protein? Do you remember how the mutation in DNA affected how our protein looked that we folded in the hallway? All right. Okay, let's jam this. Let's do it. This is the last bit of the video, right? We've done structure and function of DNA. We've done the experiments. We did replication. We just did transcription and translation. We're on to mutations. All right. Here is an amino acid. This looks intimidating because you haven't had chemistry yet. You know what an element is. So here's a nitrogen. Here's the hydrogens. You can see that there's no phosphorus, right? Remember Hershey Chase. And the R group is going to be different for each amino acid. So let's just make it fun. Here are our R groups, okay? We have Arsenal, we have the Eagles, the Chiefs, getting ready to play in the Super Bowl, Taylor Swift, Sisa, Drake. And remember what I told you in class, maybe Drake really likes Arsenal and Taylor Swift really likes the Eagles. They're going to twist the protein to get near each other. Maybe Sisa here really doesn't like water, so it's going to be towards the inside of the protein. It will be non-polar. Maybe the Eagles and the Chiefs do like water, and they'll be towards the outside of the protein. They are polar. R groups can also have positive and negative charges and want to hang out. This twisting of the protein is what gives it its shape. The Eagles and the Chiefs are stuck next to each other. They can't get away from each other, right? This is a strong bond here that I've signified. But they can twist or manipulate the protein. So if we mutated or changed something, here we change Drake to DJ Khaled. And if DJ Khaled doesn't like Arsenal, the shape of the protein may change. And so that's the big picture that I want you to get, that a change in DNA may lead to a change in the shape of a protein, which changes its function. Not always, though. So let's dive into it. This slide has a lot of text on it. I'm sorry, I ran out of steam a little bit here to make questions. So let's go through it and let's model these types of mutations. These are called point mutations. They're going to change one letter. They are also called substitution mutations because you're replacing it. You're putting one person in for slice of soccer and taking one person out. The first one would be silent, right? So we say, hey, we're going to keep the same player in. Here, the original DNA was TTC. The mutated DNA is TTT. So the C mutated into a T, right? There was an error in DNA copy. The mRNA that's going to be coded for in the original is AAG, and in here it's AAA. If you use your codon chart, these still both code for lysine. So this is called a silent mutation. It did not affect the structure of the protein. Up next is nonsense. And so you might remember in class I told you that your mom or somebody might yell at you and say, stop the nonsense. What happens in a nonsense mutation is the mRNA is mutated and it goes to a stop codon. So here, TTC, the mutation is the T, the original first T gets mutated into an A and becomes ATC. That codes for UAG, which codes for stop. The protein will be cut short. Whenever we hit stop, we stop synthesizing a protein and it's released. That's called a nonsense mutation. All right, the next one is going to be called missense. And the way I remember this is it's a mistake in the amino acid. It's going to be different. And that missense mutation may be conservative, which might mean just a little mistake, or non-conservative, which might mean a big mistake. Occasionally, mistakes can be good, right? We make mistakes and we learn. And so sometimes a mistake in a protein can make the protein better. Sometimes a mistake in the protein can make it worse. So here's an example of a conservative mutation. TTC, the second letter is mutated to a C, so it becomes TCC. The mRNA is AGG, and it's going to code for arginine. It's a pirate's favorite amino acid, arginine. Notice how arginine looks similar to lysine. It's not the exact same. You can see some different stuff up here, but the bottom looks a lot the same. That's going to be conservative. It might work about the same. It would be like if in slice of soccer, which is coming up here at Tazen, that you substituted somebody that's very similar. Non-conservative is going to be a big old change. So here we go from TTC to TGC. Notice how we're still mutating that second letter. This is interesting. But instead we change it to a G. That's going to become ACG, and that's going to code for threonine. Threonine is quite different. It looks a lot different than arginine. That's going to cause a change in the shape of the protein. All right, that would be point mutations. There are other ones where we can actually change the whole reading frame and can use this following sentence to model an insertion or deletion mutation. The fat cat ate the rat. So those are all 3-letter words on purpose, right, to represent 3 nucleotides being a codon. Here, if we delete this A in fat, the sentence becomes the, and then everything shifts over, FTC, ATA, it doesn't make any sense. And that's called a frame shift mutation. We've dramatically changed that protein. This only happens if the insertion or deletion happens inside of a protein coding region. We call this an indel. Here, we're going to insert a Z. So I'm going to pick on the word, or I'm going to do this to the word fat here, right? And we're going to insert the Z between the F and the A. So it becomes the fza. And then look, it changed it all. And once again, it dramatically changed the protein. That's it, students. That's all I got. Keep rocking and rolling. Keep studying. Asking those good questions. We'll crush it in class. We're going to do biotechnology after this. It's not on the test. And then we're going to do genetics. And we're gearing up for an interdisciplinary project. It's going to be a ton of fun. Take care. Have a good one.